For centuries, Napoleon Bonaparte's ancestry was a puzzle framed by battered parish records, migration legends, and fiery political propaganda. The paperwork traced him to Corsican nobility, but whispers of lost roots never faded. Then, scientists pieced together his DNA from a rare Mediterranean haplogroup found in his authenticated hair and a living descendant, and what they discovered challenges stubborn myths and confirms some astonishing truths. But just how much can genetics really unlock about a man shaped by war, revolution, and the burning of archives? The real story changes how we understand both Napoleon and the science that pursues him. In the cool stone halls of Ajaccio's churches, parish registers once recorded the Bonaparte family's milestones with careful, looping handwriting. The names of Carlo Bonaparte and Letizia Ramolino appear in the baptismal books of the mid-18th century, their entries marking the family as Corsican locals of minor nobility. These records, preserved in the city's archives, offer a documented trail that historians have followed for generations. The paper trail does not begin on the island, though. It stretches back to the Italian mainland, to the town of Sarzana in Tuscany. In 1490, Francesco Buonaparte, sometimes called the Maure of Sarzan, left his home and crossed the Ligurian Sea, settling his family in Corsica. The reasons for this migration remain partly lost to time, but the move is recorded in notarial deeds and civic ledgers, linking the Bonapartes to a wider Mediterranean world. Through the next centuries, births, marriages, and deaths in the Bonaparte line were entered into Ajaccio's parish books. Each entry was a safeguard against forgetfulness, a way to anchor identity in written fact. Yet, the reliability of these records has always depended on their survival. In 1793, during the chaos of revolutionary unrest and Paulist riots, parts of Ajaccio's archives were destroyed by fire. The Bonaparte home itself was attacked, and flames consumed some parish registers. This loss opened gaps in the family's documented story, fueling speculation about hidden ancestors and lost origins. Some duplicates survived in church and civic offices, and experts like Dr. Francois Crouzet have judged the remaining records as trustworthy. Still, the absence of certain volumes has made it easier for rumors to take root, rumors of exotic heritage or missing links that paper alone cannot confirm or deny. Clerks and priests tasked with maintaining the lineage worked with the tools and customs of their era. Their records are not immune to error or omission, but they form the backbone of what is known about the Bonaparte ancestry. The surviving documents confirm a Corsican identity, anchored in Ajaccio but reaching back to Tuscan roots. Yet, as the gaps left by fire and upheaval remind us, even the most diligent record-keeping cannot answer every question about the past. The solidity of the Bonaparte paper trail is real, but it is also incomplete, leaving space for new methods and new evidence to step in where ink and parchment fall silent. Napoleon's sense of identity was shaped as much by politics as by personal memory. In his teenage years at the military academy in Brienne, he wrote home describing himself as a Corsican first and foremost. In one letter, he declared, "We Corsicans are an ancient race, proud and unbending." These early statements reflected the island's struggle against French rule and his family's outsider status in Parisian circles. Yet as his ambitions grew, so did the flexibility of his heritage. When he rose to power, the narrative around his ancestry began to shift. Imperial propaganda reframed the Bonapartes as descendants of Tuscan nobility, emphasizing ties to Italian aristocracy rather than Corsican provincialism. Official biographies, court artists, and even medals circulated with symbols evoking Roman and Florentine grandeur. The Corsican years, once a badge of distinction, became a background note, overshadowed by claims to a more ancient and noble lineage. Historians such as Professor Alan Forrest note that Napoleon's public image was carefully managed. He understood the political value of ancestry in a Europe obsessed with bloodlines. In private, his family continued to navigate the tension. His mother, Letizia Ramolino, insisted on their Italian heritage to counter court gossip that painted her as a provincial upstart. His sisters, Elisa and Caroline, referenced their Corsican roots with pride in letters, but faced mockery at the French court, where Corsican was used as a slur. The manipulation of ancestry was not just a matter of personal pride. It became a tool for legitimacy, wielded to silence critics and rally support. The imperial court's messaging leaned into the idea of noble Italian blood, while Napoleon's own writings from exile on St. Helena revived the theme, casting himself as a product of Mediterranean migrations. The contradictions in these narratives fueled debate among contemporaries and later historians. Was Napoleon truly Corsican, Italian, or something else altogether? With the paper trail fragmented and rumors left to swirl, the question of origin became tangled in myth and strategy. The need for an objective measure, a test that could bypass the politics of memory, grew more pressing. As the story moves from propaganda to science, the search for answers would turn from archives to the microscopic traces left behind in hair and bone. Every human carries within them a biological record of their ancestry, written in the language of DNA. Scientists studying the past often turn to two particular strands of this code: the Y-chromosome and mitochondrial DNA. Each offers a unique window into the story of a family line, but each tells only a part of the tale. The Y-chromosome passes from father to son, almost unchanged, across generations. It is a long, unbroken chain that can stretch back thousands of years. Because the Y-chromosome rarely recombines with its counterpart, it preserves markers, known as single nucleotide polymorphisms, or SNPs, that act like signposts along the paternal line. These markers accumulate slowly over time, allowing researchers to group individuals into haplogroups that share a distant male ancestor. Surnames and Y-DNA often travel together, making this tool especially powerful for tracing the origins of historic families and verifying claims of descent. Mitochondrial DNA, or mtDNA, follows a different route. It is inherited from a person's mother, passed down through the egg cell. Unlike the Y-chromosome, mtDNA is present in hundreds or even thousands of copies in every cell, making it far more resilient in old or degraded samples such as hair, teeth, or bone. This abundance is why mitochondrial analysis often succeeds where other DNA fails. The maternal line it traces moves from mother to child, generation after generation, recording a separate history that can stretch back just as far as the paternal one. Both Y-DNA and mtDNA are powerful for different reasons. Y-DNA can confirm or dispute paternity claims and map the spread of a male lineage. mtDNA, because it survives harsh conditions and is shared by all siblings of the same mother, can authenticate relics and test kinship even when direct male descendants are missing. Yet, these methods are not without limits. They represent only two threads in the vast tapestry of ancestry, ignoring the many other lines that branch and recombine with each generation. Autosomal DNA, the genetic material inherited from all ancestors, paints a broader but blurrier picture. Over centuries, this DNA becomes diluted and shuffled, making it difficult to reconstruct precise family trees beyond a handful of generations. For figures like Napoleon, born in the 18th century, autosomal markers degrade rapidly in hair or bone. The result is that most investigations rely on the more stable uniparental markers, the Y-chromosome and mtDNA, to anchor their findings. Dr. Gerard Lucotte, one of the leading researchers in the study of historical DNA, has explained that the Y-chromosome's lack of recombination is what makes it such a powerful tool for tracing direct male ancestry. Mitochondrial DNA, he notes, is often the only genetic material to survive centuries in relics, allowing scientists to compare samples from mothers and daughters, or from siblings separated by time and circumstance. By understanding how these genetic markers work, it becomes clear why scientists focus on them when investigating the origins of a historic figure. The Y-chromosome and mtDNA do not tell the whole story of a person's heritage, but they provide a reliable backbone, one that can be tested, compared, and verified, even when the written record has grown thin. Extracting DNA from centuries-old hair is a delicate process, shaped as much by the limitations of time as by the promise of modern technology. Napoleon's hair, kept in reliquaries and private collections for generations, carries not only genetic material but also the chemical traces of its long journey. Scientists like Dr. Gerard Lucat have noted that these samples are often laced with heavy metals, arsenic, mercury, and antimony, residues from medicines used during Napoleon's final years on St. Helena. These metals, absorbed into the hair, can cross-link proteins and fragment DNA, making the genetic code harder to recover and more prone to error. Contamination poses another challenge. Over 2 centuries, Napoleon's hair passed through the hands of collectors, curators, and family members. Each contact presents a risk. A stray skin cell or a modern hair can introduce foreign DNA, blurring the line between past and present. To guard against this, researchers work in isolated laboratories using protocols designed for ancient DNA. Samples are handled with gloves and masks, and extraction takes place in rooms where no modern genetic material has ever been processed. Even so, the risk of contamination can never be fully eliminated, especially when working with such limited material. The process itself is painstaking. DNA is coaxed out of brittle strands using chemical baths, EDTA to dissolve minerals, proteinase K to break down proteins, and specialized kits to purify what remains. For Napoleon's hair, 3 independent PCR amplifications were run on each sample, with 2 separate replication runs to confirm the results. This repetition helps to weed out errors caused by damaged DNA or stray contaminants. Yet, even with these safeguards, the DNA that survives is often fragmented, yielding only short stretches that can be sequenced reliably. Researchers rely on extraction blanks, control tests that run alongside the real samples but contain no hair or tissue. If DNA appears in the blank, it signals contamination in the process, prompting a review or even a repeat of the entire experiment. Published studies on Napoleon's hair have not always detailed the use of such controls, leaving some uncertainty about the purity of the results. The information that can be drawn from these fragments is inherently limited. Y-chromosome analysis, for example, might reveal only a handful of markers, enough to suggest a probable haplogroup, but rarely enough for absolute certainty. In one study, 3 short tandem repeats predicted Napoleon's haplogroup with a 76.5% probability, leaving a margin for error. Mitochondrial DNA, more abundant and robust, can confirm maternal relationships, but it too is subject to the effects of time and chemistry. The findings, while compelling, remain probabilistic, never absolute. Heavy metals, environmental exposure, and centuries of handling all conspire to degrade the genetic record. The findings, while compelling, remain probabilistic, never absolute. Each result must be weighed with caution, understood as a piece of evidence rather than a final verdict. As the story of Napoleon's ancestry moves from the laboratory to the public eye, these constraints serve as a reminder that even the most advanced science is shaped by the fragile nature of the past. In a laboratory in Paris, a lock of Napoleon's hair, sealed away since 1811, underwent a series of genetic tests designed to answer a question that had lingered for centuries. Researchers extracted fragments of DNA from the hair, targeting the Y-chromosome, which traces the direct male line. To verify their findings, they needed a living reference. Prince Charles Napoleon, a direct male descendant of Napoleon's youngest brother Jerome, agreed to provide a DNA sample. The laboratory compared specific short tandem repeats, or STRs, found in both the ancient hair and the living prince's blood. The results were clear. The STR profile from Napoleon's hair matched that of Prince Charles, confirming a shared paternal lineage. Both samples belong to the haplogroup E1b1b1c1 star, also known as EM34, a genetic marker that is rare in Western Europe but found at low frequencies in Corsica, southern Italy, and across the Mediterranean. The match was not just a statistical probability, it was a direct genetic link across 2 centuries, connecting the archival record with biological evidence. This alignment between the documented Bonaparte line and the Y-DNA signature provided a scientific anchor for the family's recorded history. The findings offered a new level of certainty, grounding the story of Napoleon's origins in both paper and code. When the results of the Leucott hair study surfaced, the scientific details quickly lost ground to the noise of headlines. Mitochondrial DNA from Napoleon's hair, his mother Letizia, and his sister Caroline all revealed the same rare 16,184 C-to-T variant, a genetic signature passed down the maternal line. In the lab, this finding strengthened the case for the hair's authenticity and provided a maternal thread that ran unbroken through the family. But outside scientific circles, the nuance was lost. Newspapers and websites seized on the rarity of the variant, spinning stories of hidden ancestry and dramatic revelations. Headlines suggested that Napoleon's maternal DNA pointed to mysterious or exotic origins, with some outlets even speculating about links to North Africa or the Middle East, despite the variant's presence in Corsican and Italian populations. The gap between laboratory caution and media excitement grew wider as the story spread. Journalists, eager for a scoop, often glossed over the technical limits of mtDNA analysis. A single rare mutation became a springboard for sweeping claims about identity, migration, and even legitimacy. Historians and geneticists pushed back, pointing out that the 16,184 C-to-T variant, while unusual, fit comfortably within the documented maternal line and did not overturn the established record. The scientific reports stressed continuity with archival sources, but public imagination was already running ahead. The episode exposed how easily genetic data can be recast in the court of public opinion, where the hunger for novelty often outweighs the patience for careful interpretation. Ancestry, for Napoleon, was never just a matter of family pride. It was a currency in the politics of his age. Professor Alan Forrest, reflecting on the ongoing fascination with Napoleon's origins, notes how deeply questions of lineage shaped both public image and private identity in the Napoleonic era. The story of the Bonaparte family was told and retold, not just in the pages of parish registers, but in the symbols of empire, the rumors of court, and the arguments of historians. Even the nickname Maur of Sarzana, once attached to Francesco Buonaparte, was less a statement about blood than a reflection of how ancestry could be recast to serve changing purposes, sometimes hinting at distant origins, sometimes simply marking a Tuscan past. Genetic research has added a new layer to this conversation, offering evidence that can confirm or challenge the stories passed down through generations. Yet the promise of DNA is inevitably shaped by its limits. The most direct answers, those that could come from testing Napoleon's own remains, remain out of reach. The tomb at Les Invalides has never been opened for scientific study, protected by law and by the weight of national memory. Researchers have made do with what is available: hair preserved in reliquaries, samples from living descendants, and the fragile traces that time has left behind. The intersection of genetics and history is both powerful and imperfect. DNA can draw lines across centuries, connecting names in a ledger to markers in a laboratory. But it cannot capture the full sweep of a life, the choices made, or the meanings attached to ancestry in a world where identity was always in motion. The debate continues, shaped as much by what remains unknown as by what science can reveal. Today, Napoleon's genetic signature links him to a Mediterranean world more complex than his legends. As DNA technology reshapes history, every new test blurs the lines between myth and fact. What we inherit isn't just written in archives, but coded in us all, reminding us that identity, even for history's giants, is always evolving. What does ancestry mean to you? Share your thoughts below.